quick introduction of Stacy. So we have Stacy Kopp visiting us today from UC Irvine. She's an assistant professor in material science and physics and bioengineering since 2019, I believe. Before that, she was a postdoc for a couple years at National Lab. Was that one
Los
Berkeley
Alamos.
National? Oh, Los Alamos.
Mhm.
And then before that, PhD from UC Santa Barbara in physics and BS also in physics from University of Arizona. So very excited to have Stacy here with us today. Thank you for coming. And we look forward to your talk.
Okay, thank you. So I'm really excited to be here today to meet this diverse community of people here and also on Zoom. And I tried really hard to make this talk really accessible to a wide community, but I'm sure there are ways that I could have done it better. So I hope it will be interactive. If there are clarifying questions where you're just lost, please chime in. Also, we do a lot of materials chemistry, so I didn't take that part out of it, but when I show you data from spectroscopy or microscopy, I'll basically tell you what it means and explain in general terms what we take away from the data. But I think that I don't want to take away from the type of science that we do. So I want you to get a flavor for that. So you can see here in the background, we're interested in making new types of materials at the very smallest scale. So our building blocks are molecules and we use them to scaffold. In the background here, you can see a silver cluster. So we're using DNA as a scaffold to direct individual silver atoms into tiny clusters that glow. That's one part of what we do. But I want to start first by framing the way that I think about the type of science and engineering that goes on in my lab by posing a question to all of you. So I want you to think about what comes to mind immediately when you hear the term photonic technologies. I'll give you a couple of minutes and then I'll ask. So if you have an idea, raise your hand. Yes.
Light-based.
Light-based, okay, yeah.
Laser beam.
Laser beams, What else? There are no wrong answers, it's okay. Okay, yeah, lenses, okay, yeah. So, in fact, some of these will appear now here on the screen, so you can see a laser beam at the bottom. We have LEDs, sorry, people on Zoom, you can't see what I'm pointing to. Things like fiber optics or chips, this is a metamaterial. So what all of these things have in common is, or many of them are made from like solid materials, or maybe the fiber is like a glass or a polymer. But they're very like clean and pristine. So you might make this, you will need to make this in a clean room. Light emitting diodes fabricated in a clean room. Lasers, generally, you need it in a very clean space. And it's something that does basically one thing very well. When I think about photonic technologies, what inspires me are the examples that we see in nature. So for example, here's the wing of a butterfly. If you look at butterflies or other types of insects that have blue or green color, most of that color is actually not coming from pigments that are blue or green, like the fabric in your dress, but it's actually nanostructures that have length scales such that they scatter preferentially blue light or green light, like these little scales or flakes that you see here on the butterfly's wing. We also have the example of the firefly. This one is really cool. So I know they're not fireflies here. How many of you have seen fireflies? Okay, actually a lot of people. So you're probably aware that what's going on in the abdomen of the firefly is there's a biochemical reaction that causes this luminescence from the firefly. But also, scientists figured out that if they looked at the abdomen of the firefly, it actually also has nanoscale structures on it that are perfectly designed so that they couple with the wavelength of light that the firefly makes in order to enhance the scattering of light out of the abdomen, kind of like the butterfly. And then they made an LED that had the same type of patterning, and it increased the efficiency of the LED by 60% just by doing that. And then I think maybe the most beautiful example of photonics in nature is photosynthesis. So what you see here is kind of the light harvesting antenna of a photosynthetic organism. All those colored things are individual chromophores, so light absorbing molecules. They're perfectly scaffolded at the nanoscale so that they can absorb light and then transfer that energy into a chemical reaction that drives almost all of life on Earth. So it's these types of really complicated nanoscale materials that are also dynamic. They're, messy. This is not formed in a clean room, and it's multifunctional. It can be stimulus dependent. All of this is what inspires me in terms of photonic materials. So, for example, focusing on this light harvesting complex in a photosynthetic organism. Here's an example from a bacterium. You can see these little things that look like coiled wires. These are proteins, and they're holding in place very perfectly these molecules that absorb light. And so if I take away the coils of the proteins, here are these individual molecules. We're looking at really small nanometer distances. And these molecules are perfectly scaffolded, even with angstrom scale resolution, so that when light gets absorbed, that energy can be funneled to the point where chemistry happens to drive light. And if we compare this to something like a solar cell, which is very good at what it does, we generally think of those types of materials as being built out of atoms, right? So you might have a crystalline type of semiconductor like silicon. All of the atoms basically are silicon with some dopants. And we're thinking about the way that these atoms are arranged. But nature generally builds materials out of molecules instead of atoms. So it's kind of a different paradigm of building materials that differs from a lot of the commercial technologies that are available today. And this is the kind of thing that inspires my interest in designing and building new types of materials. I call it both bioinspired and bioeclectic, so I'll explain that in a minute. But I love these examples of complex multifunctional materials that I see in nature, and they're built with, you can think of them as programmable molecular building blocks. So proteins have a code of amino acids, and that controls the way that they create nanostructures. So could we use this same type of principle to make new types of materials? So in order to do this, my lab joins 3 areas together roughly. So we use tools from nature, whether we take molecules from nature, we call that bioeclectic, or we're inspired by nature and use synthetic molecules that behave similarly. We also use tools from nanochemistry and material science, and we join that together with high throughput experiments in machine learning, because at this point, the fundamental understanding of these types of materials is often really far behind where we need to be to have actual design rules. And generally, this falls into 2 categories. We're using DNA as a tool to make nanomaterials that are not biological, and I'll describe that to you. Or we're inspired by biology. We want to either replace things about natural biological materials that are maybe not advantageous, or use synthetic molecules to mimic things that nature does all the time. And so we also use block copolymers because they're more scalable than nucleic acids or DNA. So here is a little bit about me. I think it's helpful for you guys to know this. I'm from Albuquerque originally, so I grew up in the Southwest and I love the mountains. I spent a ton of time as a kid hiking, backpacking, camping, etc., mostly in like this part of the world. And then I went down south to the University of Arizona, where I got degrees in physics and math. And at the time, I was working mostly in labs that did biophysics and laser physics. So that time of my life was really about learning how to understand how nature works. Then I went over here to UC Santa Barbara, where I did my PhD in physics, and the specific research topic was in nanomaterials and photonics. So I just love things that make or manipulate light. It's one of my long-term life obsessions. But this part, I think I began to transition just from an interest in understanding how nature works to how can we create new things. And then that led into my postdoc at Los Alamos National Lab, where I worked on bioinspired photonic materials. And then finally, I've been at UC Irvine now for 4.5 years. Our mascot is the anteater. It's very weird. But these are the great people that I'm really privileged to work with. My lab is really diverse. You can look on the website and see the backgrounds that people come from. And I've already told you, we use these types of molecules. We consider them programmable. DNA, of course, has a sequence that controls its chemistry. Block copolymers are kind of like a a cruder version of things like DNA and proteins in the sense that they also have an ordering of certain chemical groups on a molecule. It's just in bigger blocks. And then we're using DNA to make these types of silver clusters, which are fluorophores. We're interested in their biomedical imaging applications right now. We're also interested in taking advantage of the way that DNA interacts with certain metals to fix issues with using DNA as a building material and potentially enable bioelectronics applications. And then we also use block copolymers as molecular Legos to put together nanoscale materials, which are otherwise floating around in the liquid. And then by organizing them in a certain way, we want to get new kinds of physics that maybe makes light emission more dramatic, for example. And I already mentioned that we use a lot of tools from high throughput experiments in machine learning because often theories aren't yet developed to help inform us design these materials. And in addition to these three areas that my group works in, because of our interest in machine learning and data mining, we've also had a number of really cool and unexpected collaborations in things involving even like metal alloys and understanding their microstructure, collaborating with metallurgists, or collaborating with people that study biomimetic electronic materials. So it's been really fun to explore new areas in that regard. So what I'm going to do is I'm going to tell you a series of stories about the type of research we do and hopefully give you an idea for my vision of what my lab is really excited about and where I hope to go. So I'm going to start first by introducing DNA, this concept of using it to build materials. Let me first show you or remind you what DNA is. Don't worry if you haven't taken biology since high school, because neither have I. So here's this double helix structure of DNA. You may remember that in this structure, we have adenine pairing thymine. I'm just going to call it A and T. And we have guanine pairing cytosine, or G pairs to C. This is a very predictable and controllable binding. So in addition to keeping all our genetic information safe, since the late 70s, early 80s, people have started to realize that this could be a building block for materials that will suddenly, we call it self-assemble, like spontaneously bind together to form different 2- and 3-dimensional structures. Why is this? Well, we see not only do we have this really programmable molecular code for how these things stick together, but we also have very defined structural aspects. So the diameter of this is about 2 nanometers, very predictable. It has a helical turn of 3.4 nanometers, also very predictable. So this is called the field of DNA nanotechnology. It's developed really dramatically over the last 20 years, especially. People have designed ways to predict what DNA sequence you need and which molecules you need to throw in a test tube so it'll bind together and form something like this DNA nanocube. These can be used as logic-based carriers for drugs, for example, or to organize different nanoparticles and cool geometries to give you some interesting light response. There's a group in Northwestern who's developed what they call programmable atom equivalents, where they coat particles with DNA and then use this very programmable base pairing to organize those particles in cool ways, basically mimicking the way that atoms arrange in solid state materials. And you can even make these over relatively large areas. This is 2 microns there, so it might not seem large to you, but for devices, that is reasonable maybe. So this is a really cool building block for organizing stuff on the nanoscale, and it's hard to find a molecule that can do exactly this at the moment. But there are some drawbacks to using DNA to build materials. One, it's really not that thermally, chemically, or mechanically stable. And that's partially because nature has to open up the double helix to, genetically transfer that information into the proteins that make up all the machines in our body. But that means if you have a gentle tapping mode atomic force microscopy, where you come in with a tiny tip and you go like this and very gently map out a surface, even that gentle touching can rupture a DNA nanostructure. Or if you have nanoparticles, which might have some electrostatic charge, like, your hair, staticky, that can rip apart the DNA origami. These things are also very sensitive to what is the composition of the solution. So, for example, if you reduce the amount of magnesium cations by an order of magnitude, they'll fall apart. So you might be able to see here in these microscopy images some of the shapes that are in the cartoons above, but below it just looks like mushes. And then finally, it's just DNA. So there's lots of things DNA can't do, and it would be great to combine the types of programmable assembly with other types of things, like maybe making it a conductor or having it emit light. So we want to be able to expand this chemical design space. And one promising way to do this is to use something about DNA that's naturally happening. So it turns out DNA has an affinity for binding to certain types of metal atoms, specifically ions. So two that are really notable are mercury, which is toxic, so I'm not going to talk about that one if I want to work on biomedical stuff, or silver, which is not that toxic to humans and eukaryotes, and other types of metals maybe to a certain degree. But if we could organize the metals inside of the DNA duplex, we can, for example, make the duplex a lot more stable. So just by sticking one silver ion here in this duplex, they were able to increase what's called the melting temperature. So the temperature at which this duplex will start falling apart, basically by 8 degrees Celsius, just one single silver atom. There's also some exciting ways to do this with making non-natural DNA bases, where you basically attach a completely unnatural molecule to the DNA, and it can do a bunch of cool things like conduct electricity or be magnetic. I'm not going to talk about that today. Okay, so this brings us to the first project I want to share with you, and that's we want to use this affinity of silver ions for DNA to make DNA nanostructures better. So how do we do this? Well, there was a study that came out about a decade ago that showed that if you have DNA strands, just the single strand, not the double helix, you can actually get this double helix to form without any of the Watson-Crick base pairing, just by adding silver. And the rules of the pairing appear to be cytosine will pair to cytosine if it's given enough silver ions, guanine will pair to guanine if it's given enough silver ions, and thymine will pair to adenine. This is kind of weird, but there are nitrogen groups on a DNA nucleobase. So for the chemists here, the silver has an affinity for those nitrogens, and then you can form a coordination bond that will bring the two bases together. Now, these things were found to be highly stable. So what am I showing you here? Well, this is what's called a circular dichromosome spectrum, which is the way that the DNA interacts with circularly polarized light that is either left-handed or right-handed. And DNA is a chiral molecule, so it has a handedness, just like my hands cannot be superimposed on one another, just by translation. So this is a really useful tool for measuring the molecular structure of DNA. So if you see that a circular dichromosome spectrum looks like another one, it means that the structure is really the same. So they found that if they heated these things up to 90 degrees, which is basically almost boiling water, the structure was not changing at all. Whereas if we compare this to a Watson-Crick duplex of 6 cytosines, 6 guanines, that thing wouldn't even be stable at room temperature at these same conditions. So silver is putting the DNA together in a way that's much more stable than what you can get naturally. You can also add chemicals that really like to denature DNA, and it will still resist to some degree this denaturation if silver is present. So what do these things look like? Well, here are some theoretical models. There are a few crystal structures as well that suggest this could be correct. But we can see, for example, here that here's our Watson-Crick duplex on the far right. And compared to this Watson-Crick duplex, these types of silver-mediated duplexes they look a lot more rigid, so they're being predicted to be a lot more rigid. This one is more elongated. And we also found that the way that the DNA strands pair goes from an anti-parallel geometry to parallel. So it's changing the rules of how we pair together DNA. So what we do in the lab is we really want to be able to understand this type of silver-mediated base pairing to the same precision that we understand Watson-Crick base pairing, so that we could use it to build these types of programmable nanomaterials that are a lot more stable than what we have just using what nature gives us. So the way that we create these is really simple. I can teach all of you to do it in an afternoon in my lab. We just mix some DNA and a silver salt in aqueous solution, and then we may or may not heat it up and then cool it down. And then we do a bunch of different types of characterization. So the three main go-tos for us are this circular dichromosome spectroscopy that I just told you about, where this is acting like a molecular fingerprint of the structure. And by comparing the way that this spectrum changes or doesn't change, we can learn about how these molecules are evolving in their structure. We also do a lot of mass spectrometry. So this is maybe a lot of chemistry. It's okay if you don't want to absorb all of it right now. But basically, this lets us count up the number of DNA strands and silver atoms that are in these types of complexes. And then the final thing is we do microscopy. And what we're using is atomic force microscopy. So that's that tool that I said you go along and touch things very gently with a nanoscale probe to map out topography. We do it in liquid, so we understand actually the real native structure of these. And we also use bioelectron microscopy, and I hope you can kind of see the stringy things over there. So with this, we want to build going from a molecular structure to a nanoscale structure, understanding of how DNA pairs together with these silvers. And one of our recent projects is we wanted to focus on this guy here, where we pair together G strands, only containing Gs with silver, compare it to the Watson-Crick duplex. We wanted to test if that theory prediction, coupled with some gas phase predictions, that these structures were a lot more rigid was actually true. Because when this is in water, its properties change a lot. And we also wanted to know, how does this thing form in the first place? We have no idea what's going on in solution while it's forming. So to do that, we use temperature-controlled circular dichroism, so we can monitor that spectral fingerprint as we're heating up and cooling down the solution. And what we see, for example, for this guy, is this dashed line is no silver added. As soon as we add silver, I have a dumb animation there, the spectrum changes immediately to this black line. So we know that means just adding silver is changing the conformation of this duplex, or excuse me, of this complex. But then as we keep heating, we see that this spectrum shifts even more, and we can monitor certain wavelengths. For example, we could look, it'll show up at 280 nanometers, we see this is continually deepening. Here at 242, there seems to be sort of a minimum sweet spot here, and this is in fact where we found we only needed to heat up to 50 degrees to fully convert this these DNA strands with guanine from their without silver conformation into the duplex. Then we can also compare this to what happens without silver. So there's this sort of sinister thing that happens when you just have guanine in solution with no silver, which is it loves to form something called a G quadruplex structure. Kind of looks like this. It's been implicated in a lot of types of cancers and other types of genetic issues. But from a building stuff out of DNA standpoint, this thing is super annoying because it's really hard to get rid of. And so, for example, we see as we heat this thing up to the same degree, it barely changes. So adding silver has a huge effect on what the DNA is going to do. Then we also found that we can make longer wires out of these DNA duplexes. So just the single G20 that I showed you, these will make something that should be about 7 nanometers long. But if we add an excess of silver, what we see is we get actually longer things forming. So this is about 40 to 50 nanometers. They have a height that's 2.5 nanometers. This is an AFM image where we're making a topography of our structures. And so this did not happen. Well, here's another image so you believe that this is real. I'm not just showing you one example. This doesn't happen when we don't have silver. So the silver is really important. And in fact, an excess of silver is important to get this structure. We don't know exactly yet what is the molecular structure, like atom by atom of this, but we have a theory that maybe when you add extra silver, it's starting to either bundle these things end to end, or maybe we start getting sliding of the Gs that are paired into different silvers, making longer wires. And this could be an exciting way to potentially test the electronic properties of these if we want to use them for bioelectronic interfaces. So finally, the last thing we did in this particular project is I wanted to know actually how stiff are these little molecules compared to nature's duplex. But how do you measure the stiffness of something that's only 7 nanometers big? So I had the wonderful privilege of collaborating with Hongbo Zhang, who's a surface scientist at University of Alberta, and he's an expert in what's called surface forces apparatus. So he had a paper where they attached either single-stranded DNA or double-stranded DNA to a gold surface, and then they have mica surface. Why? Because it's really flat, atomically flat. And what they do is they squish the DNA on the surface, and then using various types of models for how molecules behave when you squish them, they're able to get out things like the nanomechanical strength. So in our case, this is the data that they got. Let me walk you through it individually. So here is this G quadruplex case where we don't have any silver, so we know it's going to form likely some kind of wad. And in fact, they did find that this thing matched a model where the DNA would be sort of coiled up as you squished it. And they can even tell how far apart these things are on the surface. So they're 3.9 nanometers apart on the surface. Whereas if we add silver and also the complementary guanine strand, we still see the same spacing of the molecules on the surface. We expect that because it was the same surface, so that's good. But now, instead of this squished up model, it actually fits a rod-like model. And so that suggests that indeed these things are getting more elongated, they have this particular length, and then they can pull out mechanical properties from the slope of this curve. And so by doing that and comparing it to Watson-Crick duplexes, they found that these silver-mediated duplexes were about, 30%, you can read the stiffnesses here, 0.8 millinewtons per nanometer versus 0.6 millinewtons per nanometer. So this quantifies how stiff these things are. And this is really important for when we're weaving DNA into different shapes on the nanoscale. We need to know how it's going to bend. So this could be an important parameter for designing these types of nanomaterials. So I just said that, so I won't say it again. But why are we interested in this kind of thing? Well, there have been a few studies from other groups. So what's on the slide is work by other people that inspires me. One was a study of conductivity across DNA duplexes. So this was called a break junction measurement. It's basically a tiny device where you have gold at either ends, and then you measure current going across whatever is in between. So for regular DNA, this is what this group measured. Now, if they paired these things by silver, they got an order of magnitude and a half increase in the conductivity. So that suggests, okay, adding the silver is actually making this thing a better conductor. It's important to note the way they did this, it's hard to interpret their results because they didn't at that time know about silver also swapping the orientation of the strands. So they may have forced it into a less preferred orientation. So it'd be really interesting to know if you actually let the DNA do what it really wants to with silver, is this going to be an even larger enhancement? So that's one thing that inspires me, potentially making DNA a better conductor of electronic charge for use in bioelectronic interfaces. And then another thing I think is really promising is some of these groups that put together nanoparticles with DNA and order geometries, like I showed, they have started to use silver to stabilize their materials. They may not really, broadcast it, but it's being in this trade information. And it seems very important for making them much more mechanically stable. So here are some nanomechanical tests where they're basically squishing these types of materials and figuring out what are their strengths and deformation processes. So I think silver and DNA, as niche as it sounds, is actually really exciting for dealing with some very serious issues in the way that we make DNA-based nanomaterials at the nanoscale. And that's what motivates me going forward. I'm really excited to see how far we can push this. Now I'm going to transition to talking about what else we do with DNA, which is making tiny clusters of silver atoms that glow different colors for bioimaging applications. So here, I first want to tell you what is a nanocluster. So a nanocluster is the tiniest possible nanoparticle you can imagine. I call it an embryonic metal. It's so small that we have countably few metal atoms. Think about 10 to maybe 100 metal atoms. You can see some beautiful geometry, at least I think they're very beautiful, of various types of gold clusters here. What's really important to make a nanocluster is you have to have something protecting the surface, because otherwise they want to glom together and make bulk gold, or depending on the metal, they may want to oxidize. So the way that we choose what goes on the surface dictates in large part the size and shape of the cluster. And that's important because size and shape at the nanoscale gives you cool properties. So for example, here are 4 different gold clusters stabilized using different ligand chemistries, and they glow different colors. There's a bunch of exciting applications for these, from sensing and catalysis to photonics and biomedicine. If we want to use them in biomedicine, many of the types of molecules people put on the surface are really not biocompatible. And it's a big challenge to make them biocompatible. So why don't we try using biomolecules to begin with to coat the surface? And you can imagine now we can use DNA with silver, which is exactly what we do in the lab. So we take the fact that DNA likes to stick to silver ions, and then we use this to our advantage. We do a chemical process called reduction to force the silver to start making silver-silver bonds, which turns it into silver nanoparticles. But if the DNA is there, it will keep these things really small at the cluster scale, and we get these beautiful colors, this glowing. Now, what really made people excited about this in the beginning was what glowing color you got in the tube was dictated by the sequence of the DNA molecule you stuck in there. So it's kind of acting as a genome, if you will, for a nanocluster, and people thought maybe we could program the properties of these clusters by using the DNA sequence of the tool. So let me show you just how sequence programmable these are. So here's a scatter plot from high throughput experiments I have done, where I'm showing you what color on this axis. This is the color that you would see by eye, and I false colored some of these points. And then these things will be near infrared emission. So just by changing DNA sequence, we can get colors ranging in this whole spectral region. And almost all of these, except like about 10 of them, are only 10 base DNA sequences. So I'm only changing the code and not even the length of the molecule. It's a really big chemical space to play with. They have really great properties as fluorophores. I won't dwell on that, but in case you do photonics, you can believe us. Now, people got really excited about these in the first decade and a half after their discovery, and started developing all these cool applications, maybe sensing DNA mutations or toxic metal ions, bioimaging, and then some interesting photonics applications. But all of this was, in some sense, a bit premature. So the field kind of stalled, and that's because we really didn't understand the design rules for making these clusters to begin with. So you had to choose the DNA sequence that would do this cool thing, and there was really not an understanding of how to do that. So that's what drove my interest in these types of materials. So one part of the spectral space that my lab is really excited about right now is the near infrared. So I said I like things that glow. Ironically, we're working on things that glow that I can't see now. But initially, like 20, this is from our 2021 paper, there were not that many data points out here. And why is that important? Well, we're really interested in more of these because in this tissue transparency window, we call it, our biological fluids and tissues absorb, scatter, and emit way less light. And so there's a big interest in developing things that are biocompatible, small, bright, and could be used as contrast agents for light-based imaging, instead of having to go get an X-ray, CT scan, or an MRI or something. What if we could just use near infrared light, which is heat? It's not that hazardous. Now, right before I started my faculty position, my former lab mate actually discovered that there were nanoclusters hanging out here, and we just weren't looking with the right eyes, if you will. So he used, instead of a silicon-based detector, what's called an indium gallium arsenide-based detector, which is sensitive, much more sensitive to near-infrared light. He scanned one of these well plates I had designed by machine learning, which I'll tell you about in a bit, and found that there were all these near-infrared emitters just hanging out there. We hadn't been seeing them. So this was really exciting. And then there was another really exciting development the year that I started as a faculty member, which is we got the first pictures of where are the silver atoms in the DNA strands. So through a process called X-ray crystallography, other groups, not me, solved the structure of several of these. You can see them here. The DNA strands are the things that look like ribbons. These are the silver atoms. In this case, they couldn't quite resolve where the DNA was. But this was really exciting. The issue is these are still to date basically the only pictures we have. And the chemical space for DNA is huge. So how can we create design rules? Well, let me first just emphasize one more time the challenge. So if you have 4 natural nucleobases and you can have L of them in a string, this is 4 to the L possible sequences. Most people use L equals 10 to 30. So even at the lowest range, that's 1 million possible sequences to screen. It's a lot. There's a really complex connection between DNA sequence and color. So even though I've been working on them a long time, I can't, with really good predictive accuracy, pick a sequence. And at the time, there were no models. There still are very few models that we can use physics and chemistry-based techniques to answer this question. So this is what motivated me to start doing high throughput experimentation combined with machine learning. So in my PhD, I developed a way to synthesize these in high throughput with a robot and then screen rapidly the optical properties of each of the clusters. So this here is a fluorescence emission spectrum. I false colored these so that the peak is roughly the color you would see by eye. So this is what gives us information about the glow of the clusters. Now, what we do in the lab is we have a really nice robot now. We can do 384 different DNA sequences in a well plate in one experiment in an hour. And then we can scan the optical properties in the visible and into the near infrared. We also do the same type of very atomic precision chemistry that I showed you with the silver mediated base pair structures, but we do it with the clusters. And then finally, we combine all this information to try to use machine learning to map DNA sequence onto color. So let me tell you two stories about how we do that. Here's roughly an outline, and I'm going to talk a little bit more in detail about the machine learning because I think maybe this community can appreciate some of that. So we start with the high throughput experiments. We're doing 10 base DNA sequences because it's a really short length, so there's only 1 million possibilities. We can easily sample about 1,000 of them. That's 0.1% of the possible sequences. Next, we start with what we know about the clusters. So we use the information from the crystal structures, I'll show you in a minute, to define the problem that we're actually asking the machine learning algorithm to solve. And this part was really important to do this informed, like chemically informed machine learning, because otherwise it's really hard to just say, okay, algorithm, take a sequence, map it onto a spectrum, because we don't have like billions of data points like Google does on our Netflix behavior. And then we use really simple machine learning models. I think I'm not doing my animations, sorry. We use really simple machine learning models, one, because we don't have that much data compared to Google, and two, because we can use tools from feature analysis to actually learn what did the machine learning algorithm learn? What does that tell us new about the science of these materials? So here's one thing that my student Peter, who's been leading this project, found was really important. The way that we give the model our DNA sequence matters in terms of its ability to learn sequence onto color mapping. So the most common way to do this in bioinformatics is what's called one-hot encoding, where you have a string of your biomolecule sequence, and then you'll convert that, in the case of DNA, into a 4x(n) matrix that'll tell you which of these letters is in which position in this string. So basically, the result was we could never get good predictive accuracies for this approach. So this may be because we're data limited. If we had like a, all million, we could certainly learn the mapping, I'm sure. But what Peter figured out to make this work was he looked at that crystal structure that I showed you earlier, and he saw some patterns. He came to my office really excited, and he said, Stacy, I noticed that there's 2 A's at the top and bottom of this. It looks like it's like pinching it. And also I noticed that most of the sides of this are either like neighboring C's or neighboring G's. And so our thought was, okay, maybe what's really important for the way the DNA folds around this, because it looks like it's folding, is giving it information about pairs of nucleobases that are separated by some number of wildcards, which we call _n. So when we convert this 10 base DNA sequence into all 144 possible nucleobase staple motifs, then we were able to start getting machine learning models that could actually predict with good accuracy sequence onto color. The other part that was really important was related to subsampling and like statistical sampling of the data. And that's because if we look at the color distribution of our data, it's not uniform. We've got a lot of green emitting clusters, a lot of red ones, not that many near infrared ones. These peaks come from what we call magic numbers. There are certain sizes of clusters that are a lot more stable than others. So what we chose to do is we chose to divvy this up into different categories that we call color classes based on what we know about the chemistry of the clusters. And then we verified it with k-means clustering. We're also including a lot of negative examples. Most DNA sequences will give you nothing interesting at all, or really crummy chemical yield. So we include a dark class that we want to avoid. If we make a histogram of these colors, we see we have very few near infrared examples. So this is a data imbalance problem, and if our model wants to learn the difference between far red and near infrared, we need to deal with this properly. So what we did was we developed what is not a very elegant, but was actually an effective ensemble of support vector machines, SVM. And we're here, say, comparing dark versus green. So we'll take all 196 dark greens and then randomly sample 196 darks, train one SVM to distinguish that pair, and then we do it 10 times. So we have 10 different dark green SVMs. Then we do that to every pair of colored classes, and we get an ensemble of 100 of these classifiers, and we use their consensus to make a prediction about color. We can, of course, do things like tenfold cross-validation scoring. So here are the average tenfold cross-validation scores where we hold some of the data back from the model, it doesn't see it at all, and then we test how well it worked. And we would get accuracies ranging from 93%, which was great, down to 63%, which is not great, but that's the part where we have the most data imbalance. We have the least information about this part of the chemical space. So the upshot is this worked really well. What I'm showing you here is the fraction of sequences in our training data, which is like the very light colors, to the solid colors, which are our designed sequences. So we were able to amplify in all of the 3 design cases we chose, the color class that we wanted. We were super excited about this one in particular, and simultaneously we got rid of almost all of the dark sequences that gave us no interesting products at all. And you can see the color histograms over there on the right. We got a lot of things about that dashed line, which is at 800 nanometers. So that's decently out into the near infrared. And right now we're studying those in terms of their possibility for bioimaging. And then finally, I feel like am I running short on time? I don't have time. Okay. Finally, what I'm going to tell you about is a new discovery that was very surprising about the chemistry of one of these clusters. And this is also super important for their potential for bioimaging. So this was another thing I was really excited about when I first started my faculty position, which was this new crystal structure that came out. So this one, it's a really bright near-infrared emitter, not too far into the near-infrared, but it's about 700 nanometers. And it had been discovered in one of my high throughput experiments as a PhD student, so I also had a tenderness about it. But I get no credit at all for this part. This was a group in Copenhagen. They solved the first crystal structure. And what they saw were there were these 16 silver atoms, which are in gray, 2 DNA strands, the red and blue things. And then there were these 2 silvers of low occupancy. So these often appear in crystallography because what that means is in these repeating units of the same structure in the crystal, probably only 30% of the time do you have these metal atoms. This is a common thing. So we thought, okay, this is really cool. What if we took this to the mass spectrometer and we figured out how many of these silver atoms still have their valence electron? Because that's the one missing key for theoretical physicists and chemists to come along and do theoretical modeling of the electronic structure and figure out why does it glow. So I tasked my second PhD student, Anna, to do this. She started out in January of 2020, so you can imagine what happened next. But once she was finally able to get into the lab, however long that was later, she came back to my office with this beautiful mass spectrum. I guess it was probably my Zoom office, but it was still very exciting. What I mean here is, so what a mass spectrum shows us is the mass divided by charge, that's M over Z. And we have something that has a charge of 4 minus. We can also get the same thing that has a charge of 5 minus, so that's why these peaks are sort of look like they're repeating. But basically we could tell what's the mass of this thing, and there were not that many peaks, so we know like this is definitely our product. But then things got crazy because she zoomed in on this peak, and these things are basically more sodium and ammonium adducts. It's really common in mass spectrometry. We saw no integer number of silver and DNA strands. This was really frustrating. At first, I thought she was doing something wrong, of course, as the advisor, so I asked her to do it again. She kept getting the same thing. So fast forward a long time, what we figured out is these 2 things that were assigned to these silver atoms of low occupancy weren't silver at all. They were 2 chlorine atoms. So these things are chlorine. turns out chlorine, if you look at its electron density, which is what you're measuring with X-ray crystallography, it has about 30% of the electrons of silver. So it was a lot of chemical sleuthing to figure this out, but basically, once she assumed that those were chlorine, she got a beautiful match of her experimental and calculated mass spectrum, which are the red lines. And then she could also figure out that this cluster still had 6 valence electrons, and fast forward a bunch of other chemical measurements to prove this to ourselves and our collaborators, we were able to collaborate with a really great theorist in Finland who did simulations first of the bare cluster. And what he found is that if you assume that these mystery atoms here were chlorine instead of silver, it matched a lot better with the experiment, whereas the blue things that are flying away are if you assume it's a silver atom with the charge that we measured. So that wouldn't be compatible. And then they finally did the like true whopper of a calculation that took like 1 million CPU hours at a supercomputer in Finland to actually predict what would be the absorption spectrum of our cluster. Here's the experiment, here's the theory. We get qualitative good agreement, and they later did a more refined model that got really good quantitative agreement too. So this was really exciting, and this group has started to explore why do these clusters emit light? What's actually going on even at really short timescales in these clusters? And this, I think, will help us design much better clusters for bioimaging. But sometimes it helps if the scientific community gets along really well. So it turns out the guy who solved the crystal structure is also my collaborator, and eventually we were able to convince him to go back and refine the crystal structure, assuming they were chlorines. And he did do that. He got a wonderful match. That's all you have to take away from this here. But it basically showed absolutely these have these chlorine ions, which are also protecting the surface. And in the process of this whole project of detective work, chemical detective work, we found that these things are way more stable in biologically relevant solutions and buffers. And so we started screening in high throughput more of these clusters, and we found another one in time for the paper that also had chlorine ligands. We since have a few other examples that didn't make it into the paper. But these, we think, these silver clusters that have both the DNA and the chlorine ions are going to be the ones that we want to be designing for. So that's something we want to do in the future together with the machine learning. I want to show you some exciting preliminary results. So these are studies in mouse with that same cluster. You can see the glowing here in the thigh and in the tail vein. These clusters, sorry if you don't like animal images, you can look away. They're just sleeping. They died later. But for the 3 hours that we were allowed to keep them on the microscope, the glow basically did not diminish. There was some spreading around because it's going to move around in the vasculature. But I was honestly utterly shocked by this because I did not think that these things wrapped in DNA would be that chemically stable in animal. So that was super exciting. Another super exciting thing, I'm going to skip that and come back to it. Another super exciting thing is some work done in this Copenhagen group to attach this cluster to various biomolecules like insulin, which you see on the bottom right. I'm just saying that for the people on Zoom. They were able to say label a cell that's overexpressing an insulin receptor. So we can now turn these things which just glow into actually like sticky stuff that would be directed contrast agents. For example, in my lab, we're working on attaching it to things like aptamers, which will bind to stuff in the body. And all of this is related to my vision of developing nanoclusters as a new type of contrast agent for biomedical imaging. They have really a lot of promise for many of these aspects. We want to have all 6 of these things really, and we lack something that can do that right now. So I'm super excited that we got some recent funding from the Chan Zuckerberg Initiative to explore this, and this will also allow a new collaboration with the group at Northwestern who studies pharmacokinetics and biodistribution and develops new types of drugs. So they can really help us do animal experiments because I have no desire to do that myself and I wouldn't be good at it. But I'm really excited about this new collaboration. Okay, and then this brings me to the end. I'm going to wrap up very briefly on what we do with block copolymers. Don't worry, it's just a few slides, and then I'm happy to take your questions. So I want to bring us back to something we saw at the beginning, which was this light harvesting portion of an organism that does photosynthesis. So when I introduced this to you, I talked all about these molecules that I call chromophores that absorb the light and funnel the energy into the part that does chemistry. I left out, and people usually do leave out, one massive player, which was the lipid membrane, and that's all the stuff that's, you know, insensitively colored like peach here, but it's super important. So what this is a lipid membrane. Lipids are fatty molecules that half like water and half don't, and so they form these phase separated layers, and this is like super important. Without this, our cells would not exist as they do. So people are also interested in mimicking this type of behavior to create biomimetic materials, and this has been done for several decades with polymers, specifically block copolymers that have this type of water-loving, water-hating behavior, which we call amphiphilicity. And so there's been much work over several decades to make different types of nanostructures that mimic what nature makes. What I'm interested in is, can we take these types of nanostructures? So here's another example. These scale bars are anywhere from like 100 nanometers to a micron. So you can see really beautiful nanostructured behavior. What if we could now use these types of things to organize nanoparticles, which may have some cool light emitting behavior, scattering behavior, catalytic behavior, magnetic behavior, into custom geometries. I'm not an artist, so this is my bad picture of it, but we can envision all kinds of different geometries, and we might be able to then architect nanomaterials that could make and manipulate light in new ways. I'm really interested in the solution phase, so I'm not talking about block copolymer bulk self-assembly for folks who are polymer people. So one system that we've been working with is in collaboration with folks at Los Alamos National Lab, who are excellent at making colloidal quantum dots. Basically, they can make these things which glow bright red with really nice monodispersed diameters, and the surface is coated with molecules that hate water. They're apolar. Then we use the same polymer that you saw on this slide, so the same types of polymers, which is a polystyrene, black polyacrylic acid. The polystyrene hates water. The polyacrylic acid loves water. It's what's in like diapers, feminine products. It absorbs water readily. And we use this to self-assemble new types of nanomaterials. So for example, if I have the quantum dots, which don't like water, in a vial, and I start dripping in water, the quantum dots hate water so much they will crawl out, and you can see them glowing here. But if I have a polymer, the polymer will protect this and then keep it from doing this trawling. And so what is actually the thing inside the solution? Well, we've been fortunate to collaborate with experts in cryotomography at the Molecular Foundry. And what they found is that in some cases, we can actually get these really ordered geometries of quantum dots. This is not that many quantum dots. It's about 50. They're the things that look like garbanzo beans. So this is reconstructed from cryo-electron tomography, which is basically using, instead of light, using electrons to do imaging. And then just like if you get a CT scan, in that case, they tilt the detector all around your body. In this case, we tilt the sample because it's really small and it can't complain. And then we reconstruct an image, and by that I mean they did. So they found this beautiful geometries of things. They look kind of like soccer balls in the way that the quantum dots are ordered. And so we're excited about this type of behavior that it's just spontaneously happening in the self-assembly, and using this type of order to organize these particles on the nanoscale. So this goes on for a little bit, but I'll keep going. So my vision for this is, okay, now if we have a way to organize in ordered little packets of polymer, individual nanoparticles, we could start to create architectures on the nanoscale that are predicted by theorists to do cool things with light. For example, if you have a gold colloid and you put some quantum dots that will glow around it, there are certain geometries of this that are predicted to be useful for lasers or for what's called superluminescence, so a lot more light out than you would expect. Then we also have these types of things. They are going to have geometries that are like tens to hundreds of nanometers. This will get us into what's called structural color. So think back to that butterfly I told you in the beginning. If you have hundreds of nanometer scale patterning on the butterfly or like the firefly abdomen, this can give us structural color effects. And then we could also do it on surfaces to gain access to iridescence. So if we could control self-assembly across length scales by using this hydrophilic, hydrophobic interactions of these molecular and nanoparticle building blocks, then we could start to construct nanomaterials that do cool things we can't do easily from clean room fabrication. So I'm just gonna end with my long-term vision of where I see this going. I really want to understand these molecules, either the ones that nature gives us or the ones that mimic the rules that nature gives us, in order to build new types of nanomaterials with the same precision that you might imagine Lego blocks. So it would be amazing if we could make things that are just as complicated, dynamic, and beautiful as the things I've told you about. Here's another cool example of iridescence in this fish. It's a tetra, so some of you might have them in your fish tank. That iridescence also comes from nanostructure. And inspired by nature, I really think that we can address critical issues that face humanity, whether it's better ways to do medical imaging, ways to deal with energy efficiency, how to interface between our bodies and electronics, and maybe energy efficient ways to generate light. So that's my long-term vision for my research program. I want to end by thanking all the people who do the wonderful work that enables me to be here. I've got a really great group of students. Jamie is not in this picture because she joined recently, but I'm also really proud of our initiative to include undergraduates and community college and transfer students. So, for example, Josh was a community college student who worked with us remotely over COVID, which was kind of a cool thing COVID enabled him to do. He contributed so much. He's a co-author on our ACS nano paper, which is just a huge achievement for a community college student. And I also have great collaborators. I can't do any of this without them. Obviously, I'm indebted to the funding agencies for giving us the money. And then my last little bit, if you love nanoclusters and you want to know more, which might not be many people in this audience, I'm co-organizing a symposium at the Materials Research Society in December, and I'm also one of the 2 chairs of the Gordon Research Conference on Atomically Precise Nanochemistry in 2026. So with that, I'll wrap up, and I'm very happy to take questions.
I'll ask one.
Yeah.
You mentioned when you were talking about the limitations of DNA
Yes.
self-assembly. It sounded like your main complaint was about like the stability and having it be, resist, different environmental stressors, temperature changes, maybe mechanics, et cetera. Is sort of like cost and scale a problem at all still, or or is it like pretty okay to get a lot of DNA?
It depends on what you want to do with the DNA. So I'm not claiming you should ever code an airplane with it, for example. There are people who disagree with me, but I don't think that's a good idea. In terms of like the biomedical contrast agents we're working with, certainly DNA is actually a lot cheaper than some of the chemical synthesis processes, like total synthesis of fluorophores or contrast agents that are used in the clinic. So I actually think we could be cost competitive. Also for that, you need like really tiny amounts because you're working with fluorescence. But that is one motivation for why I don't only work on DNA. Because I think if we want to make really scalable nanomaterials, we have to use things like polymers. So for like specialty biomedical tools or maybe nanophotonic devices that don't require like huge scalable amounts, DNA is actually a really competitive tool. For making like a solar cell, I don't think you should do that with DNA. Some people do disagree with me. But we can order a custom DNA molecule tube that's like way more than what we need for lots of experiments on this for like $4 from a company now. So it's pretty cheap. Yes.
So I noticed that one theme throughout your presentation was like, utilizing silver, right? So with injunction with DNA and et cetera. So it's a bit of a naive question, but like, are there other like, I guess, materials or elements besides silver that can have similar properties that you just discussed? I wasn't sure if you addressed it in the talk, but
I didn't.
Okay,
and that's not a naive question. It's an excellent question. So usually people ask me, why not gold? People are like obsessed with gold. Well, there's some chemical reasons. Gold clusters are generally not that fluorescent. So if we want the fluorescence, silver is one of the most exciting metals to work with. Silver and DNA also have that special interaction where it will really only stick to those bases. whereas a lot of metals also sort of non-specifically stick to other parts of the DNA. Other types of metals that do this more specific sticking are mercury, which I have no desire to work with because it's toxic. And to some degree, copper and potentially platinum and or palladium. Beyond that, we start having to deal with the metal going places we don't want. But one thing we are exploring, I didn't mention today, is making alloy nanoclusters. So could we maybe put some gold or copper, platinum atoms inside our silver cluster? Why would we want to do this? Well, there's some work from the nanocluster community in general that has started to do this, and they've gotten really cool and unexpected properties, like maybe putting one gold atom in the center of a bunch of silver atoms, it'll become way more fluorescent. So I think that's a really exciting design space to play around with, and lots of potential. Thank you. That's a really good question. Yes.
I think Carson might have asked a similar question, but I understand that your motivations are like potentially biocompatibility and also self-assembly. So I'm just curious, like why DNA and micelles? Like why not a different structure? What kind of drove you in that direction?
Okay, by the micelles, you mean the polymer-based
Yeah.
stuff? Okay, yeah. So, sorry, is your question about the DNA or the polymers?
It's like what, when you're making the decision to work with a material, like why have you picked these two?
Oh, okay, yeah. So, the interest in DNA for me really comes from the fact that there are really not that many sequence programmable or sequence specific molecules that we have at our disposal for programmable self-assembly. So I think organic chemists are working on like really cool sequence defined polymers. This is still an active area of research, but right now it's not a tool that I can use. For nature, we have proteins and peptides, which have about 20 amino acids instead of 4 nucleobases. They can do amazing things. Obviously, our bodies are here because of that. But the chemical space is like extraordinarily big. And it's right now the field of peptide and protein, like artificial design, is much more nascent than DNA. So also maybe as a physicist, I kind of like the simplicity of DNA because it does have a limited chemical space, which is favorable for these metal particles that are very fluorescent. And then in terms of why block copolymers, well, there we also get self-assembly properties. So I think that's really promising. And also the polymer industry has been around, so long. You can like generate vats of this stuff. Yes, it is mostly based on fossil fuel petroleum products, so that's an issue, but other people are working on more renewable sources for polymers. So I think in terms of like scaling up these types of nanomaterials from, okay, we made a proof of principle thing that does this awesome thing with light, to like, actually, we want to make this into a better solar cell. You really need something that you can make in like large volumes, and that's what polymers do for us.